So the click ligation technology that we are developing, or we've developed, is kind of the chemical equivalent of ligase enzymes, which do the same job in biology. Ligase enzymes have evolved over millions of years to do a fantastic job at joining together very large pieces of unmodified DNA and RNA strands to make cells work. Chemical ligation is a bit different. Chemical ligation can do some things that ligases can't do. It can work on chemically modified DNA, it can work on chemically modified RNA, it's very good at working on very tiny pieces of DNA and RNA, which enzymes don't normally see in vivo, so ligases aren't very good at that. And importantly, the chemical method can work on a very large scale under a variety of different conditions. Another point is that chemistry is much more portable and it's much more robust, so you can actually develop kits which contain chemical reagents, and that's much more difficult to do with enzymes because transporting them, expressing them in large amounts is a problem. So there are things that chemical ligation can do that enzymatic ligation can't do, and we need to really focus on those things. We shouldn't also forget the importance of RNA. RNA is really, this is now the age of RNA. People are all the time discovering new functions of RNA in biology. siRNA, microRNA, SNO-RNA, and the ability to make heavily chemically modified RNAs and join them together by click ligation to put into cells and look at their biological functionality is really interesting. And this is part of the general area of synthetic biology. I'd like to finish just by describing 2 or 3 applications of click ligation that we have thought of and that we're working on. One really obvious one is gene synthesis. Theoretically, we can use click ligation to join together small pieces of DNA and RNA strands, and we can build genes. And the nice thing is that because we're not relying on enzymes, we can build genes with lots of chemical modifications in them that might switch on or off the gene at different times during the cell cycle. We can also make tiny pieces of DNA, which are cyclic but also form duplexes, and they're called decoys. And they will sequester enzymes like transcription factors, and therefore they have potential in vivo to switch off genes, and that means they've got potential therapeutic applications. Now these are fairly straightforward applications of click ligation, but we're aware that throughout the world in various laboratories people will have lots of problems that they can't address using conventional biochemical ligase-based methodologies. Now we can't think of all these applications. We are very much focused on certain things, and it's a great opportunity for us to find out what ideas people have, what ideas you have for using this kind of click ligation technology in many areas of biology and even in nanotechnology. So that's what we would like to hear from you.